Brian, are you in the witness protection program this morning? You're going by the name Steve Roy.
I hadn't noticed. I haven't. That's one of our hosts. He sent
okay.
this. All right, I'll change it.
That's okay. I mean, I got to think of something catchy too. I have to.
Good morning, everyone. We'll get started at just a couple minutes past the hour and let everyone join.
Hey, Max. Hi.
Morning, David. Nice to see you.
Good morning.
Hi, Judy. You look very galactic there. I like your background.
So we'll get started here in just a minute. I know there's still more people joining.
It's like waiting for the popcorn pops to die down.
All right, so we'll go ahead and get started now. So thank you so much, everyone, for taking time out of your busy schedules to join us this morning. My name is David Fleck, and I'm founder and managing partner of Freeflow. And I'm joined today by my partner, Kevin Barrett. We founded Freeflow. If people could make sure to mute when they join, that'd be very helpful. Thank you. We founded Freeflow just over 2 years ago to focus on translating basic research out of Caltech. And now we've done that with 20 companies to date, actually, in just 2 years. And so we're excited, honored, and humbled this morning to have 4 of those founders to tell their journeys and their stories about how they translated their science out of labs of Caltech into successful startups. We'll start out with hearing some words from Tom Miller, who's co-founder and CEO of Entos. Then we'll hear from Brian Stoltz, who is co-founder of both 1200 Pharma and DNATwo. Next, we'll hear from Tatyana Dobreva, who is co-founder and CEO of ImYoo. And then we'll have a discussion with Caltech President Emeritus, Nobel Laureate, Chairman, and co-founder of AppiaBio. And then we'll conclude actually with time for questions, both from us and then also from the audience. And then for the questions, we just ask that you use the chat feature within Zoom, and we'll get to as many of those questions as we can in the latter half of this discussion. So with that, I want to turn it over to Tom to tell you a little bit more about Entos. Tom?
Thanks, David. It's great to join this panel. It's great to join the event, and thank you for organizing it. I am Tom Miller. I joined Caltech as an assistant professor in 2008 and went through the tenure process there and had a wonderful research program there, focusing on the use of computational methods to describe chemical and biological processes, developing algorithms to bring more physics into the description of complex chemical and biological systems. And over the course of my time at Caltech, many of the tools that we developed were increasingly translational in nature. They increasingly led to broader collaborations within academia. They led to broader collaborations with industry. And it was clear that they began to present more of a translational opportunity. And Caltech allowed me to go on sabbatical during the course of my sabbatical to launch a company on the basis of some of the new machine learning methods that we had developed. And that was really the start of Entos in 2020. And for the first year of Entos, we focused on using machine learning techniques that would predict chemical properties across lots of different domains, materials applications, catalytic applications, pharma applications. And we used that first kind of 9 months or so to really prove the software, prove the algorithms, not only in our hands, but in the hands of customers and partners. And that led to a really, and that was of course launched with a seed round that included the Freeflow as one of our 2 seed investors, which was a fantastic beginning of that relationship. In that first year at Entos, we decided that this was an opportunity to bring those now proven tools in-house and to use them to put our money where our mouth was in terms of developing actually better molecules to address problems in oncology. So with Series A that we completed in July of last year, we again with wonderful follow-on from Freeflow and our other seed investors, we had the opportunity to build an internal drug discovery team that would be the ideal complement to our machine learning technologies, to combine those machine learning technologies with high throughput experimentation, both in terms of synthesis and in terms of biochemical and cellular assays that would allow us to drive the design, make, test cycle very quickly and efficiently. And that's led to really exciting progress over the past year in terms of fast progress in both the areas of breast cancer and lung cancer for 2 of our lead target areas. So it's been a really exciting journey. It's been very fast, and it's been a wonderful learning experience for me as a scientist. And it's been, I think, founded in what Caltech creates as scientists in the way we think about science. And I think it's been enabled along the journey in terms of how Freeflow embraces that founder vision and offers you the flexibility through that journey to achieve and to follow the opportunity where it lies. So thank you for giving me the chance to summarize that.
Thanks, Tom. Brian, I'd love to hear a few words from you. Or
Yeah, sure. I actually have a couple of slides, so
yeah,
I'll share some of those. Hopefully you can see that. So thanks David and Kevin and Steve for organizing this. Hopefully it should be an interesting discussion. So I'm here really representing a company called DNATwo, and that's a relatively new company. We founded it sort of in the midst of the pandemic, and I'll talk about that in a minute. But I thought, based on what David asked about, thought I would start with a little bit of how did I get here, where we're at the moment. So I started at Caltech as assistant professor in 2000, and I would say one of the first meetings I had, within weeks of arrival, was with the staff of what I think at that point was only called OTT, but the Office of Tech Transfer and Corporate Partnerships took me out to lunch and essentially sort of acquainted me with the way that Caltech does things in terms of thinking about how to protect intellectual property. That was literally within a month of being at Caltech. Shortly thereafter, my great late colleague, Bob Grubbs, who had at that time founded a company called Materia, local in Pasadena, asked me to join their SAB and become involved with some of the synthesis problems that they were having at that time. So it was already a sort of an immediate jump into, working with startups and thinking about things of that nature. Again, in the first year I was here, I had an opportunity to meet a guy named Steve Quake. He was an applied physicist at Caltech, at least that's the department he was in. He gave a talk, one of our general lectures, and was praising synthetic chemists. I'm a synthetic organic chemist, and I had never heard someone outside of my field say anything nice about us. So I sent him an email and said, I thought this was great. Thanks for saying nice things about organic chemists. And he responded, and we went out to lunch, and this was the kind of place that Caltech was. It's very and is. It's a collegial place where you meet people within a few months of that interaction, we were actually working with Steve on developing probe molecules, I guess you'd call them. And eventually this turned into a technology for single molecule DNA sequencing. And Steve was very entrepreneurial and launched a company called Helicos Biosciences back in 2003. At the time aimed at the $10,000 genome. Anyway, it was exciting to be involved in that. And that really, showed me that it was a lot more fun, I thought, to be involved with small companies. And so over the years, I've been lucky to be involved in a lot of, small to mid-sized companies, some within the Caltech sphere, some not, and they're sort of listed there. But all the while, we were working in our group developing new technologies for synthetic chemistry and, having whatever sort of success and accomplishments come with that. But at some point along the way, we actually invented a technology that allows us to synthesize truly uniquely substituted heterocyclic rings. And heterocycles are sort of at the heart of most small molecule pharmaceuticals. And so because of this relationship with OTT, we have protected a lot of this intellectual property, and we built up a really nice portfolio of this technology. And again, OTT brought in somebody who at the time was our first entrepreneur in residence, Dave Licata, and Dave and I brainstormed for many months about how to point this technology and aim it at something that would be useful. And eventually we found a partner in Dr. Dennis Slayman from UCLA, and we launched a company called 1200 Pharma that was based on this underpinning chemical technology that we would use as a platform for synthesis, but also Dennis's pioneering biology and which is focused on clinical oncology. So that's been exciting, and that company founded in 2017, and we're located in Culver City. And I have a weekly relationship with 1200 Pharma, if not daily. That company also has a sister company called Toro Biotherapeutics. That's an antibody-based company founded based on the biology technology of Dennis's lab. So I'm also involved with them loosely as an advisor. Holoclara was another Caltech startup that Freeflow has been involved in, and I've been an advisor to that small company for a while, and now they've had a nice raise recently, and so I'm hoping to continue to be involved. And then the main point of today, the DNATwo. So this was a company that is aimed at developing small molecule orally available inhibitors of a protein called DNATwo. So DNATwo is a multifunctional enzyme involved in replication and repair. It was discovered in yeast many years ago by my esteemed colleague, Judy Campbell. Judy's a biochemist, but she's in our division of chemistry and chemical engineering, at least half. She's joint between CCE and DVE. So I've known Judy for a long time, but she came to me multiple times, really excited about this enzyme and its discovery and the possibility that this could be really important in oncology, because tumor cells are defective in DNA repair. And so because of that, they often lack all of the sort of backup repair mechanisms that they would have typically in a cell. The tumor cell doesn't have that. So if you can inhibit those that still have DNATwo, they're likely not to be able to repair themselves. And then that would be a mechanism for synthetic lethality. DNATwo has not been drugged, so it's really the opportunity for a first-in-class drug target. For oncology, we think it's possible to target multiple histologies and hard-to-treat solid tumors. And that's really our goal at DNATwo. Our team is actually, most of them are here. Our CEO is Ken Stoltz. He's on the call. Judy, of course, is the scientific founder of the company, really the discoverer of DNATwo and really the driving force behind this. I've just been fortunate to be a part of the team as a co-founder, but also as an advisor to the medicinal chemistry group. We've identified small molecule inhibitors that have at this point micromolar potency, and we're developing now hit to lead analoging of those multiple series. That's exciting, and that's likely to take shape over the next 8 to 12 months. Helen McBride, who replaced actually Dave Licata as our entrepreneur in residence, she came to Caltech and worked very closely with Judy. She's a consultant for our team. She's now retired from the EIR position, but she continues to work with our team. That's Helen there. And we have an outstanding team of scientific and operational consultants that work with us on all aspects of pushing the science forward. So that's DNATwo. And again, thanks for the opportunity to be here today, David and Kevin, and I'm happy to talk about more of this later.
Thank you, Brian. That was great. Tatyana?
Okay, I can you hear me?
Yeah.
Yeah, okay,
Sure can.
cool. All right, I'm gonna go to slides as well. All right, can you guys see this?
Yes.
Okay. All right, so I just wanted to talk a little bit about like my life path, why I started ImYoo. So yeah, I'm originally from Uzbekistan. I actually went in my undergrad for electrical engineering, and this is where I met David Brown, which is my co-founder, in a machine learning class. I ended up experiencing originally I wanted to be an astronaut. I never thought I would end up in medical engineering. And after experiencing some life-changing health issues, I'm like, okay, the system's quite messed up. I really want to go back and fix it. After undergrad, I worked at NASA JPL on Mars Cube satellites that made it out to Mars a few years back. During nights and weekends, Richard Anderson and Viviana Gradinaro let me volunteer at their lab so I can learn more biology in exchange for some data analysis. At some point, I really wanted to get my hands dirty, be in the lab, and do the actual science. So I entered grad school at Caltech. My version of graduating was to start a company, so have like an idea to start a company. I also ended up actually getting the PhD, so that was a good one too. And then we started a company called ImYoo, and we were focused on debugging the human immune system. So just to tell you guys a little bit about it. So what we're trying to do is enable better discovery by making it easy for people to collect more and better data. It seems like a very simple idea, but right now in terms of clinical trials and whatnot, there's a lot of dropouts. People don't end up making it to the site just because there's big commutes, and also the technology that's being used is very focused on single targets. So what we did at Caltech, we published a paper with Matt Thompson, is combining single cell RNA sequencing with capillary blood. So we use this little device here to self-collect blood. And so what this lets you do is lets you track expression of many genes over time at single cell resolution. And most importantly, people can collect samples when they experience adverse reactions. So like a flare-up, one of the studies that we're aiming to do is understand migraine attacks better. So take some samples when a person is experiencing migraine or experiences a side effect. Another thing that this really allows us to do is reach out to other populations that wouldn't otherwise be able to come on site. So make a more diverse database. I just wanted to kind of show what single cell data looks like. We just graduated from Illumina Accelerator, where they gave us a lot of credits. And so we were able to do a lot of sequencing and do some of our own studies. So this is about 700,000 cells. You see different types of immune cells. And what was interesting is we were able to find a B cell cluster that was specific to someone who was experiencing chronic lymphatic leukemia. And what you can do with single cell is each dot here is a cell, is look into this cluster and see what makes it different in terms of gene expression. So kind of to tell us a more precise molecular state. So that's just an example. And I just want to say thank you for everyone who's been supportive. Caltech this wouldn't have been possible without Caltech because it's not really connected to a hospital. So we had to figure out a solution of how to get our own blood. And this all just led to this. And Illumina for giving us free sequencing credits and really helping us launch off the ground, and to all of our investors. And yeah, thank you, Freeflow.
Awesome. Thank you so much. So now we're going to transition for a little bit to have a conversation with David Baltimore. David, just want to kick off by asking some questions about Caltech. We've heard from these founders about how Caltech has been extremely impactful in their journeys, and as President Emeritus. I'm just thinking about, when you think of Caltech and entrepreneurship, what do you think of?
David, you're muted.
That happens. Sorry,
No problem.
but you can hear me now.
Correct. Yeah.
So when I first came to Caltech in 1996, as president, I found a school that was ambivalent about translational work. There were some people who had developed basic science and then the translational implications of it, people like Lee Hood, who had then just left, in fact. And there were other faculty who were absolutely opposed to the idea and said that it diluted the strengths of Caltech, which are in basic science. And over the years since then, that perspective has changed. And there's now a very strong group of people, and you've heard from some of them already, who combine an appreciation for basic science and for the translational opportunities that basic science can open up. And that actually has been an important part of my whole life in science, because when I started working on the reverse transcriptase, which I discovered in 1970, I needed help with the chemistry and found it in a company that could synthesize oligonucleotides. And I worked with them from 1971 on, and actually joined that company for some time. And so I've been at the forefront of the co-development of the basic science of molecular biology and the translation of that into industry ever since it got started in the United States. And when I came to Caltech, I wanted to see if I could help develop the translational opportunities at Caltech without changing the culture. And I think we've done that. And we still have people who are suspicious of translational work. We still have people who have gone into it wholeheartedly, and we have everybody in between. So in terms of the company that I have recently helped found, AppiaBio, one of the areas of greatest interest in the commercial world today is immuno-oncology. And I have had a laboratory focused on immuno-oncology from before when the word was developed. And in particular, I had a terrific young student and then postdoc, then lab manager, Lily Yang, who over 10 years worked with me on the translational aspects of the immune system. And then moved to UCLA, where she has gone up through the ranks and is now a professor. And Lily saw an opportunity to develop a translational aspect of cells of the immune system, in particular, a rare cell called an iNKT cell. It is not only rare, it's extremely variable in its number. Some of us have many, some of us have very few. So it's a hard cell to get a handle on. And she figured out how you could grow these cells in vitro up to huge numbers in a reproducible manner. And so she and I began talking about the commercial opportunity that this presented, because you could then use those cells as carriers of therapy, in particular for immune diseases, and cancer being the central one. And so we formed a company called AppiaBio, and it's a real Caltech startup, even though it comes now from UCLA technology, because the CEO is JJ Kang, who was a graduate student in chemistry at Caltech, and who then developed a knowledge of venture capital by working with a great venture capital group in San Francisco, the Column Group. And so JJ became the CEO of the company. Lily has stayed at UCLA and is a very close advisor on everything that goes on. And we decided to keep the company in Los Angeles, which not everybody does. And we believe that we're on the cusp of developing a really effective translational ecosystem in the Los Angeles area. And Freeflow has been an important part of that in helping companies get started that are developing in this area. And so Appia is now in Culver City. It has been through a series, very effective Series A raise. It's looking forward to a Series B raise. It's a very bad time to be trying to raise money in biotech. And so we are learning the vagaries of biotech development as well as the pleasures. I don't know if that's a quick enough
No, that's
story,
that's
but
fanta-
if you have further questions, please go ahead.
no, that's fantastic.
So I might interject here. So when I started a company that later became known as Acceleris, I went and talked about AppiaBio. This is 1984. We'd already developed the software. We already had stuff at Caltech. I wanted to give Caltech 10% of the company and continue developing. You could have probably heard Robbie Vogt yelling across the country about how professors shouldn't prostitute themselves by starting companies and doing this translation. That was the attitude in 1984. So we went outside, started, we got funding, and it all worked out very well. But it's probably better to not be at Caltech. But it was a very different attitude in '84. I think what David said about the attitude when he came is about right. The real change at Caltech was Larry Gilbert. He came to really encourage people to get IP, to start companies. He found ways to help them get started.
Thank you, Bill. And for those who don't know Bill is founder several times over, including Schrodinger, multi-billion-dollar company. So, yeah, thank you for your perspectives, Bill. So I'm just curious, David, do you feel that in some ways, do you feel that Caltech has unique advantages in creating founders perhaps because of its size, because it is smaller? I don't want to lead you too much, but are there things that you think are unique about this ecosystem now that we're really starting to see the startups come out of it?
Yeah, I think the small size of Caltech is one of its great strengths. I wouldn't want to see us try to grow into some behemoth. We do beautifully and in our exquisite way, and will continue, I trust, for forever. The environment at Caltech, because it is a small place, leads to all sorts of collaborations. You've heard about some of them in the discussion this morning. And we'll continue to do that because there are really no boundaries between the various components of the faculty. We're not tightly siloed the way other institutions are, and they're not siloed because they want to be. They're siloed because they're so big in each individual division or department that they don't need to interact. At Caltech, we have to interact in order to provide the full range of interactions that our science calls for and thrives on.
Yeah. So shifting gears a little bit, one thing I wanted to ask about was, in essence, where do you draw the line on translation and what should be translated and what should be, in effect, made open source? And what I mean by that is, just to be more specific, I know that there have been some drugs that have been developed based on research from your lab, and you haven't personally benefited at all.
Right.
But most importantly, the world has, to be clear, which is incredible. But so how do you draw the line on what you want to effectively make open source and allow others to build on top of, and what you want to hold on to and potentially create a company around?
Well, actually, it's partly just a matter of time passing. When I was a young scientist in the 70s and 80s, there wasn't the opportunity for translation that there is today. And when I made discoveries, I didn't even think about patenting them because that was not. It was not part of the gestalt of the time. Today, it's very different. First of all, the institution wants us to patent things because it wants to benefit from the exploitation of the discoveries that are made within its walls, and it's entitled to. Secondly, we've realized that when you do patent discoveries, you then can control their development, and in fact, you can bring their development along in a way that you can't if it's, quote, open source, if there isn't intellectual property protection. And so we've moved from a sort of free form development of exploitation to a much more controlled development, but I think a more effective one. And the biotech industry is an American creation. It's part of our educational system, our research system that has been just enormously effective, and I wouldn't want to see it be any other way.
One thing you might mention, David, Caltech makes it very easy to file a provisional, so you can protect your right to file IP if you need to give a talk or something. So
that's true. You can do that overnight.
Yeah, and so it's better. All the people I collaborate at other places prefer to deal with Caltech's patent office rather than their own. Much less paperwork, very straightforward.
Absolutely.
So actually piggybacking on your most recent comments, David, how have you seen commercialization of science change over time?
Well, it's gone from being a dirty word to being an accepted part of the environment. And I think the positive side of it, you can see in Tatyana's story, where young scientists have come into an environment where there's a pathway to the development of the commercial side of science. And that's, and of course, a venture capital group like Freeflow is a very important part of the system that we have in place. for funding and encouraging and learning about development from a relatively young age as a scientist.
Gotcha. No, that's helpful. What do you see as the next frontiers of translation in the areas in which you're focused? Where do you see things going? Yeah.
Well, one of the big things that's going on right now is what Tom talked about, which is the integration of computation into experimental science. Today, we don't just work in a lab, or don't just work in test tubes, but work on computers as much as we do wet lab, and actually fuse those two things in an effective manner. And I think that is a real frontier of the field. And Bill Goddard knows about that.
You're right.
Yeah, I'm happy to pick up on that. I think that really is a theme that Caltech has helped to pioneer. I think that's a great example of technologies that have led to real fruitfulness at the interface of fundamental science and physical translational outcomes with commercial opportunity. And machine learning for chemistry is a huge area that's a wonderful example of that interface.
Thanks, Tom. And let's just start to open. And we'll open it up now. And Brian, Tom, Tatyana, please just join us, and Kevin as well. Let's just continue this conversation. It's
warm already.
Fascinating.
Already warm.
One question I wanted to ask was, and perhaps to David first, but just what advice would you give scientists out there listening to this about how to think about starting a company and building a business with their science?
I think the important thing is to very carefully vet an idea to see if it is ripe, if it's ready for development. There are a lot of companies that have been formed by scientists which do very good science, but which are not really focused on a commercial opportunity. And they ultimately get eaten up or go out of business because they can't demonstrate to investors that there's any opportunity for commercialization. In doing that, it's important to work with people who have experience. Venture capital groups can provide that. They have their own interest in this situation. Our technology transfer people have been wonderful about that over the years. Larry Gilbert's name was mentioned. He really started, he's now deceased, but he really started this at Caltech just about the time I came as president. And I was very excited to encourage Larry to use his experience. And he worked with the faculty very closely and was a person you could turn to and get just terrific advice, experienced and thoughtful and understanding of the complexity of working within an academic institution.
Yeah, maybe picking up on that theme, I'll ask this to Tom, Brian, and Tatyana, to any of you, but just what made you decide that your research idea that you were focused on, that was the thing you wanted to start a company around? There's a little bit of that founder, just you have to have it's like a dog with a bone with the idea. You have to just kind of know that's the one. What made you decide that these were the ones you guys wanted to respectively
realized something.
pursue entrepreneurially?
Jack Mullion.
Yeah.
Yeah.
Happy to take a stab at that. You know, I think that at Caltech, we get really excited about, Tapio, I think, could you mute? I think that at Caltech, we get really excited about ideas that you have the opportunity for huge impact. I think that's one of the things that really defines Caltech as an institution, the fact that we should be truly ambitious in those ideas and really go after the ones that matter, because we only have, you know, so much opportunity cost associated with the time that we have. And, you know, some of those ideas are destined to thrive in academia. They're ones that will be most effective and have their full impact if deployed in an academic setting. But some of those ideas reach a stage where, you know, having the resources, having the expertise, having the duration of timescales that come with a commercialization effort is what's needed to actually execute on those ideas. To see them through to what they can become, you know, often requires a different mode of deploying those ideas. And I think that's kind of the difference of what makes an idea worth turning into a startup versus one that makes more sense to continue pursuing in an academic environment. You know, you want it to stretch its wings. Can it stretch its wings here? Can it stretch its wings there? And that dictates the choice for me.
I'll take a stab at it too, David. For us, it was actually COVID that was helpful in terms of kind of deciding, okay, we should do something about this. I mean, we were specifically sampling the immune system at a time. And after COVID came up, everybody was curious about their immune system. People started, you know, who were not in immunology or science, knowing what B cells are and T cells are. And another thing that happened with COVID is a lot of people dropped out of clinical trials. People who had autoimmune conditions that didn't want to go into the clinics because they were worried that they would get it. So we're like, okay, we could keep doing this academically, but this will have a lot more impact outside of it, and we would be able to move a lot faster. So in some ways, it's just a silver lining there, right? It's unfortunate that we had a pandemic, but in a lot of ways, it made people more aware of it, and it made the technology quite prime to put out there.
I'll also take a go at it. I would say I think a lot of times the idea of a professor sort of being the one behind a company's launch, I think a lot of times it's out of frustration that nobody else is sharing your vision that you know, or you have a great feeling for, you know, might actually make an impact. And other people say, oh, that's great. And, you know, that would be terrific, or this would be great, but they're not going to be the ones passionate enough to drive that forward. And you see the science in some ways languish from its full opportunity. I think that happens a lot. And, you know, certainly in my case with the chemical inventions that launched 1200 Pharma, that was exactly that. The pharma industry would tell me, this is great, you know, terrific, wonderful, but nobody was going to use it because it was kind of out of their ordinary line of you know, research or their ordinary path. And so you had to sort of show them. And I think, you know, it's often also the case with, I think, new targets like DNATwo. You can go and give the entire seminar and everybody thinks this is wonderful, but who's got the passion to really drive it forward? And I think that's sometimes a strong motivator.
Yeah, so actually, just following this analogous line of reasoning, or just this conversation, you know, I think for pretty much each of you, you could view it came out of basic research. So in some ways, that's almost a platform, if you will. So how did you figure out exactly? Because you have to kind of focus that when you're a company, though. You have to go after a more specific thing. You know, you can't be all things to all people, if you will. So I'm just curious, you know, David, Brian, Tatyana, Tom, again, any of you, but just how did you figure out where specifically you wanted to take it once you kind of had maybe the basic science discovery?
Well, it certainly evolves. It evolves all over the place. Please. Sorry, David.
With Appia, it just cried out for commercial development because here was a cell type that did things that have never been seen before in the combination that they're available from iNKT cells. And immunotherapy has been so successful, but in its limited way. There are many people don't get the benefit of it. And so developing cells that can bring immunotherapy to many more patients was an idea that just cried out for development and still does as we go through the stages of commercialization, which are sometimes painful.
You know, at Caltech, when we hire faculty, there's always this discussion about whether or not we like their proposals. And then it always resorts to the fact that, well, what we're going to do is just hire the best people and they're going to figure out something amazing, right? And, you know, you don't always get tenure on the back of exactly what you proposed when you came in the door. And institutions that provide that flexibility and opportunity for growth as the best ideas filter to the top in real time are strong institutions. And Caltech's a great example of that. And of course, that also happens in startups. You know, you launch a startup in the seed round, and you've got a great narrative, you've got a great story for what it's going to become. But, you know, over the course of time, you know, the landscape shifts, what work shifts, your maturity in terms of what matters shifts. And having institutions, in this case, investors, that offer that same sort of flexibility and environment to grow and to pursue what really matters as a young company is another important thing, and that inevitably happens and that strong investors can provide.
David, I'll jump in here with a question, unless I just cut somebody off.
No, go ahead, Kevin. That was
so from an investor perspective, Freeflow, you know, considers the founding team to be really one of the most important things, potentially in some cases the most important consideration about making an investment. And we like to think that we sort of, you know, mold and help shape the company around the founders. So we think about it in terms of what skill sets and what experience is necessary to really equip the company once funded with the best chance to be successful. So each of you have different approaches to this. Some of you came in with an academic partner who was a co-founder. We have some of those people. David Brown is on here today. Judy Campbell is on. But so each one is a little bit different, though. So maybe you could each just talk a little bit about the team, the founding team when you're first funded.
Kevin, can you be a little like more specific, like founding team is in terms of like, why did we choose them to be part of the team, or like
No, not why did you choose them, but what else did you need? You made, for example, at ImYoo, you made one key hire very early on, but how did you think about what do you really need to add, to be clear, in order to really maximize the opportunity to be successful as a startup?
Yeah, at least for us, it was all about like milestones. And it's kind of weird because in science, we always think about long term. It's like, hey, how's this project going to turn out in 10 years, 20 years? But here, funding cycles are like about every 2 years. So you kind of have to think about, in 2 years, will that person help me get there? Kind of mindset. Like, you know, I don't have this right now. Can I get it? Whereas, you know, in graduate school, you always think about, I'm going to do everything myself. But here is like, how can I get there faster? Otherwise, I will not have money to keep going.
You know, to me, the key hire in any company is the CEO. And if you can find a CEO who really understands the ecosystem, getting money, getting talent, getting regulatory approval, getting all the little bits that have to come together, then you've got the right kind of person. And actually, that person may not be the best CEO 5 years down the line, because by then you should be in a much more commercialization framework, commercializing framework, and you may need a different kind of person. So it's getting the person who serves the needs of the company at the time when you're hiring them.
This is Judy Campbell. I'd really like to second that. So when we had the idea and did the preliminary studies that showed that the DNA replication repair interface was the Achilles heel of many types of cancer cells, and we used all our facilities at Caltech to develop our first inhibitors that might turn into drugs. And we had enough success, so I was impassioned to start this company. However, I knew that I knew nothing about and had never had any experience outside of academia. So I started my career when there were very few women, even in academia, much less in industry. And so my first goal, and I would not have started the company if I had not been able, through our EIR, Helen McBride, to identify a very talented CEO. And he has been, Ken Schultz, instrumental in freeing Brian and me just to do the science. That's all Brian and I do. It's just like running a lab. And Ken takes care of everything else. I mean, Brian and I have done pitches in order to raise funding, but Ken keeps track of all the money, all the legal, all the financing, and a view as to when we should go for new funding and that sort of thing. So for the young people who are wanting to found companies, I suggest you get yourself a professional CEO and you really work with him or her to get your company started, because it has been instrumental for DNATwo.
So just to connect the dots for everybody, Judy is the faculty member professor at Caltech who made the foundational discovery, which ultimately led to DNATwo. She and Brian Stoltz, one of our panelists, were co-founders, and they identified Ken Schultz, who's also on the call today, who came in as the CEO of DNATwo. And there's Ken now.
I do think COVID also played a role for us. So I'm just thinking it through, maybe not so much from an immunotherapy perspective, but by just kind of bringing people together in kind of a different sort of manner. But yeah, Helen, your entrepreneur in residence, and I had gone to school together. I went to medical school. She went the PhD route, and yeah, everything just kind of came together. I mean, a full alignment, which worked out well.
Thanks, Ken. Back to you, David.
Yeah, just one question that I see in the chat is from Dan. Thanks, Dan. Is about how you identify getting to this founding team again. You know, oftentimes it's scientific collaborations that end up, you know, maybe the genesis is at Caltech, but then it ends up, or maybe it ends up with collaborations with, I think each of you actually, or most of you have, you know, the founding origin stories are Caltech, but also other institutes. I'm curious, how did those come about? Was that just organic, and then all of a sudden you decided to create a company after you had that collaboration, or do you purposely seek out those collaborators at other institutes in order to drive your company and research forward?
I'll take a stab at that. So in the case of 1200 Pharma, we had, I thought, a great chemistry technology, but we didn't know where to go with it. And it was really ultimately seeking that out, but personal connections, I think, also play a huge role in this. You know, at that time, the EIR, Dave Licata, knew on a social level Dennis Slayman at UCLA, and he sort of knew what he was doing, but then found out more. And, you know, Dennis and I connected, and that led to, you know, to this, a great collaboration. Started as an academic collaboration, but with an eye toward thinking about how to launch a company. For DNATwo, you know, it's a different scenario. We're heavily dependent on a great team, as I mentioned, of consultants and outside experts that we help to use to interface with the CROs. And Judy and I interface with those teams, but those sort of operational consultants really, you know, seek out the best CROs to partner with and work the day-to-day with them. And, again, a lot of that was, through connections. One of our, I think most important consultants so far was a chemistry friend of mine from a postdoc, and he essentially runs the entire medchem operation operationally for our company. And it's that sort of personal connection that I think each of us bring that I think plays a big role in how these things develop. But it's not easy. It definitely takes a lot of work because, different people are committed to different things, and, you have to find the right person, you have to find the right team. It's not easy, but it's kind of fun.
Los Angeles has an enormous range of talent that's tended to focus on the particular institutions where people are, UCLA, USC, Caltech. And I think there's a whole movement now to try to bring together the strengths of these institutions so that they can be synergistic. And in particular, to provide to Caltech a stronger outlet in clinical science,
Right.
and in clinical investigation. So I actually started a joint center of of clinical work with UCLA some years ago. It doesn't much exist right now, but it put me in contact with Tony Rebus, who is a physician at UCLA, works in myeloma, in melanoma. And Tony has been terrific as a contact. Also, I have an ex-student, Owen Whitty, who's at UCLA, and he has been terrific in providing contacts in the medical world. And we're now actually all working together, USC people, Caltech people, and UCLA people, to try to develop an LA center under the auspices of Chan Zuckerberg. And we have an application in right now that is developing to do that. I think that could provide a unification of the strengths of the LA basin. And even though we have this terrible problem of being separated by freeways, there are ways around it, and we're finding them all.
Yeah, I think the capabilities of Zoom prove that point. I think, just the
Zoom had, again, the pandemic has been a positive from that point of view,
Yeah.
teaching us how we can work together through the computer.
Yeah. Just looking at I'll get one more question here. I see Charles asked a question for Tom. Tom, starting with an ML platform and then building a drug discovery capability, what were some of the challenges to convince investors? Because I know you had a great A round raise, and that Entos could pull this off.
Yeah, I think basically investors want confidence you can execute on the idea, right? And they'll believe everything you've done in your past, and they might give you the benefit of the doubt on one next thing, sort of, that you haven't quite done yet. So, when I came out of Caltech, I had never run a company. So the seed round was basically, great, you're a scientist, you're proving your chops in that way. Can you actually build a software startup? And then, we execute on that in the seed round. And then, in the next stage, you've shown that you have tools in hand that are truly differentiated. You've built a company around that core idea, and you want to take the next chapter of the company. They'll give you confidence in that, but they, you take a leap of faith. And a lot of people didn't, have confidence that we could build a biopharma. I mean, you got to hire heads of chemistry, heads of biology, you got to build labs, you got to, I never had labs before. So it was quite a leap of faith that we'd be able to execute on that idea and create a high throughput experimentation thing that would be the counterpart to the machine learning. And finding the right investors to get behind that idea is really important. So, you know, and people believe what you've done. And that's why execution is really important.
Yeah. Great. Well, I know it's the top of the hour and people most likely have places to go, people to see. So just again, want to say thank you so much to David, Tom, Brian, Tatyana, and everyone who's attended this morning. Just really appreciate the support, and we look forward to doing this again very soon. We're doing these now on a consistent basis with different topics. So stay tuned for our next one. But again, thank you so much.
Thank you, David.
Yeah, thanks, David.
Yeah.
Thanks,
Yeah.
Kevin.
Thanks, everybody.
Bye,
Bye.
everyone.
Yeah, thank you, Kevin.